Wonderful. In the interest of time, we'll move on. Please join me in thanking Maria again for the wonderful presentation. There's more seats in the front if people want to move into the first few rows. Please feel free to move up. We'll continue with our proceedings presentation given by Monica Dayao from Carnegie Mellon University. And she will be talking about deriving spatial features from in situ proteomics imaging to enhance cancer survival analysis. Over to you, Monica.
Thank you. Hi, everyone. I'm Monica Dayao. I'm a fourth-year PhD candidate at Carnegie Mellon University, and today I'll be presenting my work on deriving spatial features from in situ proteomics imaging to enhance cancer survival analysis. And this is work in collaboration with Enable Medicine and the University of Pittsburgh Medical Center. And so in recent years, there's been a number of consortia whose goal is in part to create a single-cell 3D molecular human atlas. And if we were to achieve this goal, this would allow us to have an unprecedented understanding into human development, aging, disease progression, etc. However, it's quite a daunting task as humans have around 30 trillion cells. And not only do we have to understand the cells in the body, we can't just consider them individually since we're not just a bag of cells. We have to understand them in their spatial context, so how they interact with each other. And with these consortia, there's been a number of data modalities that people have been using, such as genomics and proteomics. And one of the data modality that we're focusing on is single-cell spatial proteomics data. And what that means is single-cell meaning a single-cell resolution, spatial meaning measuring the actual spatial location of cells in situ, which enables us to directly see cell-cell interactions and the organization of tissue. And proteomics meaning measuring the abundance of proteins. And in recent years, there's been a number of data modalities that have been developed that enable us to look at the abundance of tens of proteins in situ. And this is just kind of an overview of a bunch of the different modalities that exist out there right now. A lot of them are either fluorophore labeled based, DNA labeled based, or metal ion based. And at the end of the day, they all kind of look like something like this. So this is an IBEX image showing around 50 different proteins. Each color channel is a different protein. And yeah, so now we can see on the order of like 30 to 50 to 70 proteins at once, which is great. We can have a further understanding of the samples that we have. And so one of the things we wanted to focus on in this work is specifically how this data relates to patient outcomes. And some examples of this would be survival length, recurrence, disease severity. And traditionally, a lot of clinical metadata can be used to predict patient outcomes, such as age, smoking alcohol history, tumor stage, sex, etc. And there's been a lot of work in recent decades that have looked at gene expression, a correlation with these patient outcomes, such as survival. And so for us, we wanted to look at whether we can predict survival directly from spatial proteomic imaging data, and specifically find out which features are predictive of survival. And to do this, we kind of wanted to answer 3 questions. First, how do we extract features from this data? Next, does the spatial information actually give us useful data over non-spatial features? And lastly, how exactly do these spatial features correlate with survival? And so the data that we're using is a head and neck squamous cell carcinoma data set. This is the 6th most common cancer worldwide. There's almost 1 million new cases every year and 1 million new deaths every year, and this is only going to be increasing. There's a lot of traditional risk factors involved, such as tobacco and alcohol consumption, but also infection with HPV or EBV is a risk factor. And unfortunately, there's only a 40 to 60% 5-year survival rate. And so our data set is 281 codex samples from 81 patients. We have 39 proteins measured. In this presentation, I might be using the word biomarker instead. And there's 2 million cells with over 7 batches or cover slips. And we've labeled 16 cell types, which I have on this bar chart here. And on the right, this is just an example of one of the samples. And so one of the first ways we can featureize this data is just take the average protein intensity across the entire image. We can do this across the entire image or within the individual cells. And so this just gives us a 39 length vector, giving the average expression for each biomarker. Another way we can featureize this data is take the fraction of cells present in the sample. Since we have 16 cell types, that just gives us a 16th length vector that sums to 1. Note that neither of these methods use the spatial information in any way, it just looks at the sample as a whole. And so how can we incorporate the spatial information? Well, we create something that we call the neighborhood matrix. And how we do this is we take our image and then we perform segmentation and classification. And then we consider the k nearest neighbors of each cell. In our case, we use k equals 10. And then we look at the cell type composition within those k nearest neighbors. And then we form this neighborhood matrix M, where element Mij is the fraction of cell type J within k nearest neighbors of cell type I, averaged across all cells of cell type I in the sample. So each row here sums to 1, and the matrix is not necessarily symmetric. And so great, we have average biomarker intensity, cell type proportions, and this neighborhood matrix to featureize these samples. How else can we featureize or describe the spatial structure of these samples? Well, we can turn to something called spatial point patterns, in which we describe each single cell, or represent each single cell as a point. And how we do this is we take our segmented image, we look at the centroids of each of the cells, and each of these centroids becomes a point. And to each point, we can assign the vector of biomarker intensity for that specific cell. And why would we want to use this kind of spatial point patterns? Well, there's a lot of tools in this field that's already established in other areas of science, such as ecology and geology specifically. And with these tools, there's readily interpretable and clear statistical definitions available. And so one of the tools that we're using in this work is Ripley's K function, whose goal is to measure correlation or dependence between points. And this is the function. And basically what it's doing is that it's going to each point, drawing a radius r around it, and then summing the number of points within that radius. And so that's just described by this function here. We note that for a completely spatially random process, or in other words, a Poisson point process, in which points do not have any correlation with each other, this is equal to pi r squared. We can also compute a weighted version of this function, in which we basically do the same thing, but each point now is assigned a weight, and then we add those weights as multipliers in this function. But the rest is still the same, and the completely spatially random version of this function is still pi r squared. And to give an example of what these functions look like, so we have 3 different point patterns here. And on the bottom is the K function. The dashed line is the K function for a completely spatially random process. And on the left, we see that the points are a little bit more dispersed, so our function falls below that dashed line. And on the right, the points are a little bit more clustered, so the points fall above the dashed line. Note that there's a common transformation that divides by pi and takes the square root. And all that does is makes these graphs look like this, which makes it a little bit more visually interpretable. And so for our data, what we do is we segment like before, and we look at the centroids. And for each point, for each sample, we compute both the weighted function for each biomarker and the unweighted K function, just using the points themselves. And so these are the equations again. And we perform this transformation, divide by pi, take the square root for both of these. And then what we do is we subtract the weighted K function, we subtract the unweighted K function from the weighted K function. And we call this the transformed K function. And then we compute values of this function at r equals 30 and 80 microns. We chose these through some hyperparameter analysis, but also 30 and 80 microns kind of correspond to, at least in our data set, small neighborhoods of like 2 to 3 cells and larger cell neighborhoods of 6 to 8 cells. And so our final feature vector for each sample is a 39 times 2 length vector, since we have 39 proteins and then 2 values of R. And so for an example of what this looks like with our data, here's an example sample. On the top left is the DAPI stain, just kind of to give us an idea of where the cells are in the image. And on the right is our transformed K function. We can imagine as the 0 line to be, or the 0 value to be where the protein expression within the image is kind of randomly located with respect to the cell locations. And so the orange line on the bottom represents CD68, which is on the bottom left. And this is kind of indicating to us that CD68 in the sample is kind of dispersed throughout the sample with respect to the cell locations. And then the top blue line is showing the CD45, which indicates to us that there's some clustering effects of the CD45 expression with respect to the cell locations. And great, so now we have a bunch of these input features. We have the neighborhood matrix, this K function, and the cell type proportions, and we can train a survival model to predict survival. And so what we use is a random survival forest model, which is similar to a random forest, except we're going to be predicting mortality risk, which is a proxy for survival. And how we evaluate this type of survival model is using something called the concordance index, which is a score between 0 and 1, measuring concordance between predicted risk scores and actual survival length. Essentially, 1 means completely concordant, 0 means completely discordant, and so 1's better. And so what we did is we trained random survival forest models on all of our samples using 2 cross-validation schemes, one grouped by patient and another one grouped by batch, to see if our method is generalizable to both batch and patient effects. And on the left is just the patient folds, on the right is the cover slip folds. And these first 3 rows are showing the performance using the non-spatial features, so average biomarker cross image within cells and the cell type proportions. These next 3 rows are showing the feature sets that use spatial information. So our neighborhood matrix performs the best out of all of the individual feature sets. We find that the Ripley's K function on its own actually performs quite poorly, but in combination with the neighborhood matrix, it provides a slight boost in performance, which indicates to us that there might be some complementary effect of the K function information with the neighborhood matrix. We also performed some methods from previous works that have aimed to predict survival from this type of data, and we found that our method outperformed those as well. And so now that we have these models trained, can we actually use the predicted risk scores to stratify patients into low and high risk cohorts? And so what we did is we took the model trained on the neighborhood matrix features and averaged the risk scores across samples for each patient. And then separated the patients based on by thresholding on this risk score. So then now we have 2 cohorts. And what we find is that the 2 cohorts that our survival model found are statistically significant, or have statistically significant survival probability, which is great. It's showing that our model is learning something important. But what are the actual differences between these low and high risk cohorts? Like, what are the important features? And so what we did is looked for differences between in neighborhood fraction values between these 2 cohorts. And what we found specifically is that macrophage fibroblast interactions as well as naive immune cell fibroblast interactions were enriched in the high risk cohort. And so we forwarded these on to our collaborators at UPMC, and they're currently performing some experiments based on the results, but some preliminary results have shown that that head and neck cancer fibroblasts, they induce cytokine secretion, which enhances tumor growth. And so there's some confirmation that these results make sense. And so, yeah, overall, what we've done is developed a framework to extract spatial features from this highly multiplexed data modality, specifically for survival prediction. We've shown that spatially dependent features do outperform non-spatial features. And we've also found that our framework can give us risk-based cohorts and reveal features that can inform future experimental design. And some future directions, we would like to apply our framework to different patient outcomes, such as recurrence. And we want to investigate the performance of other spatial point pattern functions, as there's quite a few out there. I'd like to acknowledge everyone in my lab, as well as the people I've collaborated with at Enable Medicine and UPMC, as well as my funding. I encourage you all to check out the paper if you haven't already, and thank you.
Thank you so much, Monica. Very exciting work. I have actually a couple of questions. So it's very interesting to be looking at neighborhoods. I think it's not an easy problem. How big do we need to go in terms of cellular neighborhoods? How big is meaningful, or how small is meaningful? And also, how do you weigh your points in the Ripley's K metric?
Right. So for the first question, in terms of how large the neighborhood should be, unfortunately, I don't have the slide here, but we did try different values of K for the neighborhood function, and we found that a range between like 8 to 15 cells like gave very similar to results, whereas too many cells kind of blurred the I guess the information within the sample, and too small, I didn't really give that much meaningful information. And then the second question was, the K function. Yes. So we weight by the biomarker expression for that particular cell. Yeah.
Okay, thank you. One question there.
Hi, that was a very interesting talk. Thank you. I have a question. I guess I have two questions, but she had asked the first one. The second one, it seems like for this model you have, there seems to be a couple of steps, and I'm just curious about how your segmentation might affect your neighborhood definition, because essentially you're computing the probability that, I guess, the crossroads that each point is a neighbor of the next point to it. Do you think there is some play with how you segment your images, and how might that affect your neighborhood definition and your classifications?
Thank you for your question. Can you clarify again? You're asking so how the neighborhood is defined, or no?
I think I understand how it's defined, but the first step you do is you segment.
Oh, segmentation.
So you segment,
yes.
And then you classify yourself as a cluster of data.
Yeah.
So I'm just curious how the segmentation affects the clustering, which essentially affects the neighborhood. Because if you're losing information from the segmentation, that sort of propagates.
Yes.
That's right.
No. Yes. That's a very good question. And segmentation is a very tricky problem that has, in my opinion, has not been solved yet. We did try a couple of segmentation methods. And none of, at least the ones we tried, none of them were perfect. And so we just chose one that, I guess, visually looked the best across samples. But we didn't do too much further investigation on how it would affect like the downstream results. But I would imagine there's some potential effect depending on the segmentation method. And I think that's something that still needs to be worked on in the field.
Thank you, really interesting talk. I'm wondering about the effects of averaging the standard composition of your k-nearest neighbors, and especially if you looked into if there's some kind of sensibility to the way the fields of view were defined, because in some cases you might be like mostly centered on cancer tissues, and sometimes you might have more of the surrounding tissues, and I guess the standard interactions might change depending on what you think.
Thank you for your question. So all of the samples that I used were of like primary site tumors. I guess I didn't look specifically on like what proportion of the of the slide was actually tumor versus surrounding cells. But I did restrict my samples to be the ones only from primary site tumors, just to ensure to have some consistency there.
One very quick.
Yeah, hi. Thanks for the excellent talk. But my question is, we know that there is a lot of heterogeneity between macrophages and fibroblasts. Some are pro-tumorigenic, some are anti-tumorigenic. How are you going to address that? And clinically, we know that HPV patients do really well compared to tobacco smokers for head and neck cancer. So did you find the differences when you looked at them? All the data was smaller data set, and you found those interactions. What about multivariate analysis, given that there could be other confounding factors responsible for the poor or good outcomes in these patients?
Thank you for your question. So for HPV specifically, we did, I don't have any slides here, but we did look at whether HPV correlated with survival within our data set. And we did find that, you're right, HPV patients did have a much higher survival rate than our HPV negative patients. But we didn't incorporate this necessarily into our analysis, but we did find that our K function features did have some significant differences between the HPV cohorts, but we didn't include that in this specific survival prediction.